We're going to start.
All this and eventually the same thing.
Thank you. Richard Tehan obtained a PhD in medicinal chemistry program at Oregon State University of Pharmacy, where he studied drug discovery and chemical ecology in insect-pathogenic fungi. He is currently assistant professor of biochemistry at Utica University, where he teaches chemistry and biochemistry, conducting research focused on secondary metabolism, drug discovery, chemical ecology, evolution, and taxonomy of fungi, with particular interest in Cordyceps. It gives me great pleasure to introduce Richard Tehan.
Hello everyone, thank you so much for coming, and thank you Joe for that introduction. Tonight I'm going to tell you all about the Cordyceps of North America. I know everyone's here at these forays for all the charismatic megafungi, the amanitas and the boletes, but tonight for the next hour I'm going to make you look at a bunch of moldy bugs. Okay, so for this presentation I'm going to especially emphasize the classification, so how these fungi are classified, how they're identified. I'll go into some of their biology and chemistry and some of the research that's going on in my lab and elsewhere. Just a little background on myself. I'm originally from Utica, New York. I went to Utica University, where I did a bachelor's in chemistry. After that, I went out to Oregon State University, where I studied in the medicinal chemistry lab of Dr. Carrie McPhail. I also collaborated very closely with the mycology lab of Dr. Joey Foray. And so Carrie is kind of my chemistry mentor and Joey my mycology mentor. And after I did a postdoc, I came back to Utica, where there was a position open for professor of biochemistry. So that's what I'm doing now, teaching and doing research on fungi. And the fungi that I study mostly are this group of insect pathogens. I'm going to call them the Cordyceps. Cordyceps sensu lato, so Cordyceps in the broad sense. And that includes the formal genus Cordyceps, but it also includes, as a shorthand, it includes all of many different groups of fungi. And there are many of them. There are hundreds of species. There are, it may be in the ballpark of 2,000 described species in this group. And surely many thousands more yet to be discovered by science worldwide. They grow on all sorts of insects. They grow on beetle larvae and the nymphs of cicadae and moth pupae, and some of them grow on truffles, like this one here, has a ton of stromata coming out. Again, I'm going to talk all about the different groups of fungi and their ecologies. Okay. So let's look at that truffle parasite, for example. Here's the deer truffle, an Elaphomyces species, and here's the fruiting bodies of this Cordyceps coming out of it. It's a Tulipocladium species. And if we zoom in on the cap of this mushroom, we can see it doesn't have gills or anything like that. It's bumpy. It's papillate with the protruding osteals of the parathecia. We can see in a cross section, here's all of the actual fruiting body of the fungus, which are called parathecia. And if we look at a cross section of that with the microscope, we can see it's this flask-shaped structure with this neck. And inside of that is all of the asci. These are ascomycete fungi. They make their spores in these sacs, the asci. And so as the fungus develops and the ascospores enlarge, they get extruded out of the neck of the parathecium, and they burst under pressure and release the ascospore. So here's the ascus, with the ascospores inside. They have this thickened cap. And when the ascus bursts, it releases the ascospores, which are these filamentous spores. They're just about as long as the ascus itself. And the ascospores in Cordyceps are typically these very long filamentous spores that are multicellular. They divide several times, and they lay down cell walls, and they often break along those septi into secondary ascospores or part spores. Okay. And when these ascospores divide, they typically have a fixed number of rounds of cell division. So when 2 becomes 4, 4, 8, 8, 16, you get an ascospore with 16 with 16 part spores in it, such as in this truffle parasite. And if it divides 5 times, you get 32 part spores per ascospore. It could divide 6 times, in which case you get 64 part spores per ascospore. Or it could divide 7 times, and you have 128 part spores per ascospore. Yes, I did count each one of those spores to make sure I'm showing you 128 part spores. And I've never seen any with more than that. I think they've kind of run out of space to lay down separately. So that's these are conserved characters in each of these species when you're identifying cordyceps. Is how many part spores do they have in their ascospore? The parathecia have different ways they can be arranged. So they can either be entirely immersed within the fruiting body, so they're entirely inside of the cap of this one or this one. Or they can be partially immersed, where they're sticking part way out. Or they can be entirely on the surface of the fruiting body, in which case we call that superficial. They're entirely on the surface of the fruiting body, entirely all the way exposed. Right? So they don't always form the sexually reproductive structure, making ascospores. Sometimes we find them in their asexual state. And we often recognize that by these powdery fruiting bodies that are covered in conidiospores, the asexual spores of these fungi, such as in all of these various specimens. Okay. When they make an asexually reproductive structure on a stem, we call that a cinema, or these are various cinemata of different cordyceps. And sometimes they aren't powdery, they might look smooth macroscopically, but if you were to zoom in on these Ophiocordyceps species, you would see actually they're covered in tiny little hairs that have spores on them. So even these are just covered in spores, but you can't see them easily without maybe a hand lens. Okay, so I've been calling all of these fungi cordyceps. This actually includes many, many genera, and here's a list, maybe not even a comprehensive list, of all the genera that you could include within the cordyceps in the broad sense. Okay, we've historically classified all this group of insect fungi into the genus Cordyceps. Anything with a club-shaped fruiting body growing on an insect, we call it Cordyceps ever since Fries defined the genus in the early 19th century. But there's been a proliferation of names, and for various reasons, we now have several dozen genera, okay? And not all of them are on use. I have the ones that are in orange are the ones that we currently use, and some of the older names too, which, you know, if you're doing research, you need to know the older names too. The name Cordyceps, by the way, literally just means club head. Okay, and I don't have time to go through all the genera, we'd be here all night, but I will go through the different families. So what families are the Cordyceps classified in? Well, we used to classify them all within the family Clavicipitaceae, which is a family that was first described by, does anyone know who first described that family? I'll give you a hint, he studied Pyrenomycetes. His first name is Clark. Clark Rogerson, yeah. So the person who named this Foray is named after first described this family. He studied related fungi, not really cordyceps, but other closely related ones that also make parathecia. As of 2007, though, we split up the families into 3 different families. We have the Cordycipitaceae, the Clavicipitaceae, and the Ophiocordycipitaceae. And then a couple of years ago, we now have a 4th family, the Polycephalomycetaceae, that was split off of the Ophiocordycipitaceae. So I'll go through these families, and I'll kind of tell you some of the differences between the 4 main groups of cordyceps. And one thing I want to point out here, the one reason for this slide, is to point out a lot of people are tempted to just lump them all into the family Cordycipitaceae. Well, I don't know what genus or species is, I'll go to family, I'll call it Cordycipitaceae. But that's actually not a really good catch-all name for any of these fungi, because it only represents a small sliver of this entire group, right? So Cordyceps itself is actually just a better name as a catch-all name. So let's look at the family Cordycipitaceae. Fungi in this family are typically some shade of white, yellow, orange, or red, so somewhere on that spectrum. They often grow on moths and crickets and beetles and spiders. In fact, if you find a cordyceps on a spider, it's almost certainly in this family, because they only seem to occur in this family for some reason. There are some exceptions to that, but I won't go into those. They usually have their parathecia arranged ordinally, which is to say at 90-degree angles. They're sticking straight out of the stroma. And how else might they stick out? I'll show you another way they might stick in another family. They usually have long filamentous ascospores that break into part spores, so that's typical. Sometimes there's actually several species that instead make a different type of ascospore. They make the bola ascospore, which looks like this ancient hunting weapon, the bola, and it's connected by one long filamentous thread and two fusiform ends where all the cells and nuclei are, which is really bizarre. Why would the fungus make such a weird ascospore? But it does. No one knows why. Okay, this family also includes the genus Bovaria. This one's commonly found, and this is one of the very few generalist pathogens in this group. Most of these fungi they exhibit strict host specificity. They only occur on one insect species or one small group of insects, whereas this species grows on all different types of arthropods. And you can identify it by its mold state, where it makes these cotton ball looking conidia balls, these balls of conidia spores that just look like cotton balls under a hand lens. Okay. It also includes the acerioid fungi. Cordyceps tenuipes, for example, is this characteristic moth pupa pathogen with a yellow stem, and it looks like it's been dipped in sugar. If you find one with an orange stem, that's probably something in the genus Samsoniella, a related genus. This species, by the way, was discovered in New York State by Peck. Cordyceps also includes the family Gibellula. We found several of these today. These are spider pathogens that have these beautiful conidiophores on their asexual morph. So they have these pom-pom-like conidiophores sticking off the cinemata under a microscope. They're very beautiful. They can be this paintbrush shape, or they can be this more globose shape, or sometimes they can be both in one species. So it's not maybe not very diagnostic nowadays. So moving on to another family, the Clavicipitaceae, in the strict sense, include some of the most beautiful cordyceps, in my opinion. They come in all different colors, green and orange and purple and brown. These ones, unfortunately, aren't very common in North America, though they do occur. So I won't spend a lot of time on this family because we don't encounter them much. Sometimes, though, we do see them in their asexual state. If you find an insect that's wearing a fuzzy green sweater, that's probably the mold state of Metarhizium. When they do make the sexual state, when they're making perithecia, the perithecia in this family are often obliquely arranged at sharp angles on the stroma, sticking out at a sharp angle. You can see a cross-section here where they're sticking out at a sharp angle. And the necks of the perithecia are curved. So that's some of the characters of this family, how you would distinguish it from the other families. So I'm going to move on now to maybe one of the more common families that we find here is the Ophiocordycipitaceae. And this includes, I would say it's dominated by the genus Ophiocordyceps, which is one of the largest, I think the largest genus of Ascomycete insect pathogens. And it's due for splitting. Unfortunately, we're going to have to get used to some new names in the fullness of time. All of these species are currently in the genus Ophiocordyceps, but they will definitely be classified in other genera in the future because, well, that's already underway where we started to split them up. Now we have the genus Paraiceria. Okay, this family includes a lot of beetle pathogens, cicada nymphs, hymenopteran pathogens, ants and bees. In fact, if you find a cordyceps on a hymenopteran host, an ant or a bee, it's almost certainly something in the family Ophiocordycipitaceae. I like to think that this family of fungi are maybe more sophisticated than some of the other cordyceps in the sense that they can overcome the social immunity of the hymenopteran hosts like ants that would normally discard their sick members away from the colony. But Ophiocordyceps has a unique mechanism to get around that social immunity. When it infects an ant, it makes it climb up a tree and out on a stem and bite down on the stem where it kills it and forms a fruiting body out of the back of its neck, where it can then rain down spores on the forest floor, infecting the entire colony, getting around that social immunity of the ant colony. Okay. It also includes the genus Tulipocladium, which includes all of those truffle parasites that we used to call Elaphocordyceps and Cordyceps. Fungi in this family often have their fruiting bodies with aperithecial apices, which is to say they don't have perithecia on the apex, the tip. Right? So if you look at the tip of these fruiting bodies, there's no perithecia. They don't extend all the way to the tip. So that's a character that you commonly observe in this family, and in fact, really just in the genus Cordyceps primarily. Another one where it's only on the side, on a lateral pad, where you have the perithecia. Okay. Also, if you see a fruiting body with superficial perithecia on this wiry stem, this kind of fibrous stem, that's another character that you only observe in the genus Cordyceps. So not Cordyceps in the strict sense. On to the fourth family, the Polycephalomycetaceae. These are fungi that we often find in the asexual state with these viscous conidial heads, these kind of slimy, globose-looking things, and also kind of with that lateral pad, similar to an Cordyceps in the sexual state. Sequencing is kind of shown. We used to call them all polycephalomyces because they have these many heads. Polycephalo, many heads. So polycephalomyces is how we used to lump them all, but we now have several different genera because only some of them are closely related to true polycephalomyces. So if you find that kind of morphology, it's nowadays lumped in this bigger lineage that we call the Pleurocordyceps. So if you find something like that, we used to call them polycephalomyces. We now call them Pleurocordyceps, most of them. Okay. These fungi are really interesting. They are usually, or maybe almost always, or maybe always, hyperparasites of other cordyceps. It's a cordyceps, a parasite, growing on another parasite. Cordyceps growing on another cordyceps. You find them growing out of the fruiting body of an Cordyceps here, right? You can see there's one cordyceps, and then all these white cinemata of the Pleurocordyceps. Right? And again, the fruiting body of an Cordyceps with the Pleurocordyceps growing out of it. Right? And then another one with the fruiting bodies growing out of an insect, but alongside an Cordyceps. And maybe even here, this is just growing out of the endosclerotium of the Cordyceps. That's possible. We just don't know. Okay, so here's how these four families are arranged. Cordyceps is the sister family to the Polycephalomycetaceae, which together are sister to the Clavicipitaceae. But if you look at the Cordycipitaceae, you'd have to go back in the family tree pretty far, and here's the Cordycipitaceae, which is actually, we think, more closely related to the Hypocreales. Someone name a fungus in the Hypocreales. They're usually mushroom parasites. We probably found some today. Hypomyces. You get a Cordyceps sticker. Yeah, you were paying attention in class. So Hypomyces, the lobster mushrooms. You all know the lobster mushroom. It's a mold that grows on a russula, and it shares ancestry with Cordycipitaceae. In fact, it might be more closely related to Cordyceps than Cordyceps is to Cordyceps, right? Okay, so when I moved out to the Pacific Northwest, I very much had mushrooms in mind and thinking about these lush green temperate rainforests and all the mushrooms I would find there. But the conventional wisdom was that out in the Pacific Northwest, there were no Cordyceps to find, or they were rare or not abundant. So I didn't really get my hopes up on that. There were certainly a lot of big trees to look at while we're out mushroom hunting. There's a big old Douglas fir. Sometimes they're covered in mushrooms, like this huge Sitka spruce covered in chicken of the woods. But then I heard a rumor of an Cordyceps that occurred out at the Oregon dunes, these sand dune systems on the Pacific coast of Oregon that are dominated by this dune grass and Pinus contorta and strawberry plants, and that's about it. There's not a very diverse habitat, but a very unique habitat to go mushroom hunting. And I went to the Oregon coast every spring for 4 years looking for this Cordyceps. And it was like looking for a needle in a haystack. Look at all this expansive, dunes that just go on for miles. Dune grass, where am I going to find this Cordyceps? I just looked and looked and looked for years. Until one day, there it was. There's the fruiting body of this fabled Cordyceps. And if you dig down in below the sand, you find its host, which is the larva of the 10-line June beetle It's actually a pretty big cordyceps, as cordyceps go. There's my hand for scale. They sometimes had one stroma, looking a lot like a species we have here, Cordyceps ravenellii. But this species is actually distinguished from that East Coast species in that it almost always had two stromata sticking up out of its head like devil's horns. So 99% of them were like that. And by the way, when we found them, we found hundreds. It was pretty cool. Sometimes you'd see them with three, a mutant, and one time I even saw it with four stromata, a double devil. Okay, so there it is. They have these kind of purple fruiting bodies when they're immature, and they start out with immersed perithecia that become superficial as they mature, and they become studded with these black superficial perithecia. DNA sequencing shows that it's distinct from the East Coast species that we have, and we've proposed a formal name for it, Cordyceps satanis, the devil's cordyceps. Okay. Another cordyceps you can find in the springtime out in the West Coast is an ant pathogen that there were records of, and I went looking for it, and there it was. Obviously, look at that. It's so easy to find. That little yellow dot in the moss is this ant cordyceps. It's another new species growing on carpenter ants. That we're proposing the name Cordyceps oregonensis, although we now know it occurs throughout the West Coast, on California up to British Columbia, so maybe we need a more inclusive name. Here's our photo plate. There's a species on the East Coast that's very similar and closely related to it that also has this kind of yellow fruiting body, often found sticking up out of the moss. But this one grows on a different hymenopteran host. It grows on wasps. In fact, I found this the day after I got lit up by yellow jackets down in North Carolina, and then here's one that's neutralized by an Cordyceps. So it's very gratifying to see. They also grow on the parasitoid wasps, the ichneumoniid wasps. Okay. Another species we find out here is pretty common is Talipocladium ophioglossoides, formerly Cordyceps ophioglossoides, the golden thread cordyceps. And this one grows on a truffle. And it has this characteristic yellow context and yellow rhizomorphs. So if you find a cordyceps like that, you know you're looking at this species. But there's a similar species found in the southeast that's pretty rare, but it looks very similar to Talipocladium ophioglossoides, but it doesn't have those very yellow rhizomorphs, and it's not yellow on the inside. That's Talipocladium japonicum. That first cordyceps that I showed you, the one with all the stromata sticking up out of the truffle, I showed you the ascospore earlier too, and we didn't count the part spores. When we actually count the part spores in that species, we count 11. Which is really bizarre. Remember we said that they usually are in multiples of 2, 2, 4, 8, 16, and so on. But this one makes 11 part spores, which is a very strange character. So this is another new species found in Louisiana and Alabama. So far there's only 2 records of it, and I've given it the provisional name Talipocladium undecasegmentum, the 11-segmented Talipocladium. Another species in that genus is this really inconspicuous one you find on the side of rotten logs. There's me for scale. So get up close with rotten logs and look for this fungus. You can see there, if you look close, it's got perithecia. That's like a cordyceps and its fruiting body, but it's a much reduced fruiting body. It's not that club shape like the others. And if you dig down in that rotten wood, you'll find its host, which is the larva of a beetle. Okay? This is actually a famous fungus called Talipocladium inflatum, synonym of Cordyceps subsessilis, and it's famous as the source of the drug cyclosporin A, which was discovered from this fungus in the mold state. Mycologists in Europe isolated this fungus from soil, where it also occurs, and grew it as a mold and discovered this drug, and it's a powerful immunosuppressant that's been used as an immunosuppressant drug to prevent the rejection of foreign tissues and foreign organs. So in the 1980s, it made organ transplantation something that became possible. Before that, it was really experimental, but this transformed medicine. And later on, it was found that this mold is the same fungus as this Cordyceps. That was a discovery that Kathy Hodge made in Ithaca, New York. Moving on to another beetle pathogen, a much bigger one, Parasiria insignis. This species here's a specimen that Jay Justice sent to me. Maybe some of you know Jay. He found this growing in his backyard in Arkansas. This is one of the biggest Cordyceps in North America. This is 13 centimeters top to bottom. And it's growing a very large longhorn beetle larva. And he sent it to me. I got really excited thinking maybe it's a new species. Spoiler, Cook and Ravenel beat us to it by a century and a half. But I got really excited. I got it in culture and made some chemical extracts and tested those. In a bunch of the assays, we were just testing, hundreds of samples on at a time. I threw this in the batch, and we found that this fungus inhibits protein secretion in cancer cells and was very toxic to cancer cells. So it became very exciting to me overnight and part of my dissertation to study it. And I started sourcing Parasiria specimens wherever I could. There was a rumor of one that grew up in the Cascade Mountains. This one that grows on the hump-winged grig, an endemic cricket to the mountains of Oregon and Washington. And we gave that a new name, Parasiria cascadensis. And Carrie McCabe sent me, and others sent me some really excellent cicada pathogens, another related species. We gave the name Parasiria pseudoheteropoda. From the cricket pathogen and the beetle pathogen, I isolated 8 new compounds that are super toxic. These are the compounds that were killing those cancer cells and that we're interested to study as anti-cancer agents. And we just, on Tuesday, submitted our paper for publication. You can read the preprint on ChemArchive. So that's available. So we're studying these compounds as possible cancer drugs. We're really excited about their activity. We found out that they inhibit the production of proteins in cancer cells, inhibiting translation, a lot like the cancer drug plitidepsin. So it's all well and good to discover new chemical compounds and try to figure out what they do in the laboratory and see if they are therapeutic, if they do anything to cancer cells. But I want to know, what do they do in nature? I wanted to go back to the specimen and ask, why does the fungus make these compounds? And in order to do that, I set out to do a molecular cartography experiment to map out where are the compounds, where are these toxins made in the host specimen system, the host parasite system, right? If they're made in the aerial portions of the fruiting body above ground, the fertile portion or sterile portion, maybe they're anti-herbivory agents, keeping things from grazing them and eating them. But maybe they're made in the rhizomorphs below ground, where they're growing through the soil, interacting with the soil microbiome. Maybe there they would make antimicrobial compounds. But maybe if we could find them in the host, we could propose that they are virulence factors, targeting their host, inhibiting host immunity, and so on. Right? And the host, by the way, is filled out with a fungal endosclerotium, so the fungus has completely consumed the insides of this larva. So my question here is, where do these toxins occur? Do they even occur in this native system? At this point, I only know they're growing in the laboratory culture. So I performed mass spectrometry to see if we could detect these compounds, and yes, we get a signal, definitely when I map these compounds out in the specimen. But the question then is, where in the specimen do we find these? Do you think we find them in the host, or in the rhizomorphs, or in the, excuse me, I pointed to the wrong thing, the host, rhizomorphs, or the fruiting body? Does anyone want to take a guess?
Host.
The host? You get a sticker?
Oh, I get a sticker. Thank you.
You're welcome.
In fact, yes, we find these toxins almost entirely inside of the host of this fungus, right? So these compounds have some ecology directed to, or necessary for being inside of the host and not the fruiting body. So we can now begin to form hypotheses about what they might be doing there. And so one other thing that came out of this study was I was reminded of a case series I read where 60 people in Vietnam were poisoned after eating cicadae that were infected with what was identified as Parasiria. And there was even one fatality. The people eating those cicadae were, after the insect. A lot of people eat insects, and they found these insects that had fungus on them. They thought they could just collect the fungus, they obviously saw the fungus. They thought they could chop the fungus off, keep the insect, and still eat that, right? They even cooked the cicadae before eating them. So the question arises, would cooking neutralize these toxins? Could these toxins be what sickened and even killed people? And so I set out to destroy these compounds by cooking them after months of my life trying to isolate them. So I heated them up to 175 degrees Celsius. Right? That's 350 degrees Fahrenheit for 30 minutes. And they were completely unchanged. That's what this figure shows. This is the NMR spectrum before heating and after heating. It's the same spectrum. The compounds are unbroken. They're unchanged. They are super stable compounds. Keep that in mind. That's going to come back up. So now that I'm going to publish these compounds, I need to say what fungus they're from. And I thought I had all new species. I couldn't identify them. They didn't match to anything that was common. And so I went to go through the literature to try to see if there were any matches to descriptions of fungi that match these Cordyceps, these Paraceria species. And I looked at the old names, all the Cordyceps. There was 587 records of that, and 307 records of Ophiocordyceps that Paraceria used to be in that genus, and 17 in Paraceria. All told, about 900 species that I had to go and look and make sure didn't match any of these species before I gave them a new name and embarrassed myself. And there was one description that I read of Cordyceps insignis collected in 1880. It was collected in 1881, and it was collected by Ravenel in South Carolina, and he sent it to Cook at Kew in London, and he described it as livid purple, and I almost dismissed this and said, next, this isn't the one, mine's not purple, it's brown. And the last thing they said, though, stuck with me. They said, but it reminds us of Cordyceps capitata, now Talipocladium capitatum. And I gotta say, these beetle pathogens Jay Justice sent me reminded me of Talipocladium capitatum. So they look very similar. They have that kind of similar fruiting body. Well, there's no way to tell. Microscopy isn't very useful in this genus, and so on. So I had to settle it. I loaned the type specimen. There it is, the original specimen that Ravenel collected in January 1881, a sad, shriveled specimen dried and glued to a piece of paper. And I tried to sequence DNA for this 142-year-old specimen. But you can imagine a century and a half does number on DNA. And the DNA was too degraded, and I couldn't sequence DNA to compare them genetically. But can you think of maybe another molecule that could be present in this organism that is very stable? The Paracerium, yeah, these toxins that I'd spent years studying. I decided to sample it and see if we could detect those toxins in this holotype specimen. And I knew right where to sample. I took from the endosclerotium, the host, right? And analyzed them by mass spectrometry and definitely detected them. It was, in fact, an exact match to this beetle pathogen that Jay collected in Arkansas. And in fact, distinct from the cricket pathogen that we also studied. And so to put a finer point on that, I went through all the Paraceria species and specimens I had on hand, 5 different species, 21 different specimens, and compared them all at mass spectrometry, and showed statistically we can make them group into their phylogenetic clades only with the chemical data. So thereby we can take an older barium specimen and link it to a phylogenetic clade with the chemistry. Okay, so here's my photo plates of those species. We published that a couple of years ago, the new names for them. Okay, and I'll just finish with a few more species I'll tell you about. Another Paraceria I studied in that paper was Paraceria monticola, which was collected in Tennessee in 1936 on a mole cricket. Here it is. And this one hasn't ever been found since, only known from the holotype. There's actually several Cordyceps species like that in the USA, lots that have been collected, described, and forgotten. And I call them the lost of us. And then there's another one out in Washington State that was also collected by Smith in Mainz. And Mainz named it Cordyceps washingtonensis. It grows on Lepidopteran larvae. It looks like Cordyceps militaris, he says, but it's pinard yellow, so some shade of yellow. It has non-disarticulating ascospores. So if you ever find yourself out on the West Coast and find a yellow Cordyceps, you might have found the rarest Cordyceps on Earth, only found with the type collections in the 1940s. By the way, it was collected on the shores of Baker Lake, which was subsequently dammed, and the level of the lake has risen up 100 feet, so the type locality is probably underwater, in fact. Okay, so the last thing I'll tell you about is a few suggestions for how to go about finding cordyceps. I hope I've inspired you to think about cordyceps and maybe try to go find some yourself, because they're actually a lot more common than most people think. So my tips for finding cordyceps are the 4 L's: logs, limbs, leaves, and look in creeks. You gotta get up close and personal with those wet, juicy, decorticated, rotten logs. If you see a rotten log like that, slow down and look close, because you might see a tiny little thread of larvae like to go and burrow in these logs, and then the fungus infects them and forms a fruiting body out of the log. Here's Cordyceps stylophora, a pathogen of click beetle larvae. Another rotten log. Here's another Cordyceps. 1, 2, 3, 4. If you look up close, that's Cordyceps variabilis, a pathogen of fly larvae, and it pigments them orange with the fungal pigments. Has that lateral pad with the aperithecial apex, classic Cordyceps. Here's another picture of the same species turning the host orange. Gotta look in those rotten logs. Limbs. Some species you typically or only find along limbs. So you gotta scan the stems and look for the zombie ant fungus, Cordyceps kimflamingiae, which this is probably just north of its range. I don't think we find the zombie ant fungus here, but there might be other ones. I've been telling people to go and look inside rotten logs. There's some northern latitude ones occurring in rotten logs. And Cordyceps clavulata, a scale insect pathogen. And here's an undescribed Cordyceps growing on the Carolina leaf roller cricket. Okay, number 3, you gotta flip over leaves. I spent a lot of time today doing this, looking for Gibellula. Here's 2 spider cordyceps in the genus Gibellula, with those beautiful cinemata with the pom-pom conidiophores. Here's the sexual state of Gibellula. It doesn't look the same, but that's the remnants of the spider, the abdomen, and all the perithecia ribbing it. Another new species, this one from Oregon, but there's a similar one that I've collected in New York. Here's a few from today. So I spent time flipping leaves today, found probably 15 specimens of this in this genus. Wow. It was pretty dry and crunchy out, but I didn't know what else to do with myself, so I flipped leaves all day and found all these moldy spiders. Okay, and you don't just find Gibellula, you find Cordyceps, Purpurea cilium, Malariella, and others. Purpurea ithacensis, discovered in Ithaca. Okay, I'm going to double dip on leaves. Also, don't just flip leaves, but look in leaf litter. You'll find a lot of moths pupating in the leaf litter, and you'll find Cordyceps there, and some other, Cordyceps militaris, and other Cordyceps species. Okay, and lastly, look in creeks. Creeks are damp. This is good for mushrooms generally, but it's especially good for the cordyceps, which often require cooler temperatures, and constant cooler temperatures, constant moistures, the right humidity, and for some reason, creeks are just a hot spot for cordyceps. Insects are drawn to creeks, and so on. And it's really good to get in there with a flashlight and look in those dark nooks and crannies, and you will find cordyceps by doing that, I promise. Okay, here's a good resource. If you do find some cordyceps, go on iNaturalist. I hope you're posting all your mushroom finds on iNaturalist, immortalizing your finds for posterity and contributing to the data we have on fungi. And if you find them, please add them to the North American Cordyceps Sensulato project. And you can see the species, the observations roll in and how many species we've identified. We've identified over 101 species in North America. And you can click on the links, click on the species and see what species we have that are common. You can click on any one of those and see where they're occurring. You can see where they're all occurring throughout North America. And the eastern USA is a hot spot. We are in a great place to go cordyceps hunting. Okay, how much time do I have? A few minutes. So, lastly, when I moved back from Oregon to New York, I moved into a musty old farmhouse. I mean, a charming old farmhouse. And I went down into the basement and looked up in the rafters and saw all these moldy spiders. And I got so excited, I knew just what that was. That's Angiodontium aranearum. I've wanted to see that species for years. They didn't even tell me it was down there. It would have been a selling point. So naturally, I got it in culture, and we grew it in the laboratory. I had one of my students do some analyses on it, Jalen Wright, and he analyzed it with mass spectrometry and detected the bovaricins, another family of cyclic peptides, kind of like the paraceriamides. This was a known family, but a known family of toxins that hadn't been detected in this genus before. In fact, Jalen was the first chemist ever to do any chemical analysis of a fungus in this genus. So kind of a new discovery.
When you grew it in the laboratory, it didn't have insects to feed on.
No, some of them will just grow really well on potatoes. We use potatoes or Cheerios, actually. I'm not kidding. Yeah, it's cheap carbohydrate, and it's part of a growing fungus's balanced breakfast. I drag my students out in the woods so we can go pick mushrooms and bring them back to the laboratory and culture them. And here's a few of the cordyceps we have, growing in the laboratory. We got lots of other fungi too. They all make interesting chemical compounds. But here's a few of the cordyceps forming fruiting bodies in the flasks. I just wanted to show those. Okay. And then lastly, here's the phylogeny of the family Ophiocordycipitaceae, showing some of those new species. There's a lot of them. Some of the new species that we're working on in the various families, Ophiocordyceps, Talipocladium, and Pleurocordyceps. So there's a lot in the pipeline, a lot of new species. Here's the 3 Paraceraria that we already published, and there's a lot more on the way, so that's a little teaser. Here's the latest photo of my lab with all my students that are working hard. Actually, some of them already graduated and went on to medical school and so on. So I just want to acknowledge all their hard work and the research we've been doing. So my students at Utica University, I still have several collaborators at Oregon State University, including my advisor and the Spatafora lab, Ishmael lab, and their students. We've had collaborators from the Kyoto Prefectural University, University of Helsinki, and funding has come from the NIH, the NSF, Oregon Mycological Society, Cascade Mycological Society, and the Sonoma County Mycological Association. And the most support comes from my wife and son, Carla and Ronan, my mushroom hunting partners and partners in crime. So with that, if anyone has any questions, I'd love to take the questions. And if you want a Cordyceps sticker, you can buy them from my website and my lab's website, and it will support our research. Question here?
Okay, because my questions run the sublime to the ridiculous. We'll go simple one. The simple one is, how do the cordyceps manage to infect the truffle underground? Like, what is their dispersal method? I understand how they get the insects, but how do they get to the truffles?
That's a great question. The question is, how do these cordyceps parasites, these truffle parasites, infect their host? How do they infect the truffle? And I don't know the definitive answer to that. I can speculate a little bit. In fact, the insect parasites infect their insect hosts in various different ways. Some of them, maybe the spore lands on the host and it penetrates with the structure in epithelium and can colonize it that way just from the outside. Others of the insect pathogen hosts, they're eating a plant, and the plant has the fungus growing inside it as an endophyte, an asymptomatic fungus, not causing harm to the plant, but it gets inside the host that way. That may or may not be how it gets into this plant-associated truffle, right? This is a mycorrhizal species, but no one knows. But one thing we do know is that the ancestor of all the truffle parasites was a cicada pathogen. So it jumped host from cicadae to truffles. And where are cicadae? They're sapsuckers on the roots of the same trees right next to those truffles. So at some point in time, this parasite, the cordyceps, found that there was this other niche that it could occupy, this juicy, tender morsel of nutrients that it could, if all the cicadae are taken, maybe I can take this truffle instead, and all those free nutrients there. So it jumped host from the cicada to the truffle. Exactly how, is it coming from the plant? Is it coming from spores in the soil or things like that? We don't know.
Okay, so here's the ridiculous now. All right, so we know, because there's a television series on cable, that the cordyceps can infect humans and make them insane. All right, but have you seen any evidence that, I mean, I did not know cordyceps could parasitize a fungi. Beyond the insect kingdom, have they gone anywhere else?
Well, I guess technically, spiders are outside of the, insect phylum. I guess the same kingdom. Several of them are only known they're only known from the soil as, free-living soil fungi or nematodes. Nematodes often in the soil. Endophytes in plants. Whether or not they have another life stage in their life cycle, they might live as a plant endophyte for a time until they find their insect host, things like that. A lot of it's just not known, but for several of them, we only know them as saprophytes or endophytes, things like that. So we just don't have the full picture. We don't have all the data to be able to say. Great question. Question here?
So when you grow them in the lab on potatoes or on Cheerios, they're willing to become vegetarians? I mean, I'm assuming that they're getting protein from the insects?
That's right, yeah. So, well, a lot of these parasites, they're not obligate parasites in the sense that they need their host to survive. They're free living very often in the environment, in the soil and in plants and in rotten wood. And maybe they're there lying in wait for their final host of their life cycle, the insect or the truffle, as it may be. So during those other life stages, they have a different nutritional mode. They're saprophytes. They're getting up organic matter out of the soil, or phloem out of the plant, or whatever it is. So yeah, they can just eat nutrients if you give it to them. So they're very versatile in that sense. Yeah, that's surprising to a lot of people that they're parasites, but they can also be decomposers, saprophytes. Yes.
So they would be like a mosquito that needs a blood meal in order to lay eggs, and for them to make spores, they may need the protein from the host.
Oh yes, yeah. So I guess it's an open question. Why do they need to have their host to make spores if they can grow on all those other things? And I've wondered that, and I've asked other mycologists. And my thinking is, it's the explosion of nutrients that they get when they do colonize their insect hosts. So all that nitrogen is the signal for them that now it's time to form the fruiting body, because now they have enough nutrients to invest in a fruiting body. Right? So in fact, they're very well timed. They have seasons. They fruit only when it's their season. They fruit only in the same stage of the insect host every time, like a moth pupa is when they've made a lot of fat, and that's the time to colonize them and get all the nutrients, maximize the nutrients. So they're very timely. They don't just virulently infect and kill as fast as possible. They time it. So it's synchronized. Or
I have two more questions.
sure.
I have an interest in Bovaria bassiana because it's used for bedbugs. And it's wonderful because you don't have to move anything. You just spray around the bed, and the bugs walk through the layer of dense spores and pick up the spores and bring that into their nests, and within 2 or 3 weeks they're all gone.
Wow, I love that. Yeah, so
So I'm wondering if there's more. I know that's been used in greenhouses before for insects for a long time, so it has a certain safety record to take it into homes.
That's excellent. Yeah, I hadn't heard that before. The question was about Bovaria bassiana. That I mentioned it earlier. It's one of the generalist pathogens. It can grow on maybe any arthropod. And so I wasn't aware of the bedbug treatment that it could be. So that's exciting.
If you're looking for it, if you ever had trouble, the product is called Apprehend with 1 P.
Apprehend
Yes.
is the name of the commercial product. Excellent. I was aware that it was used in agriculture, but I didn't know about the bedbugs.
And then, the toxin that you found have you tried that out on insects, or?
I mean to do that, because we need to test that hypothesis. Is it intended to kill insects is one open question. So we need to test, is it even toxic to insects? That's still an open question. We've only tested on human cells so far, mammalian cells.
And one last question. I hope you haven't been affected by the cuts in funding.
Sadly, I can't say that. I haven't. I have. We had a very well scored big NIH grant that went down the tubes recently. So cutting funds were a big bummer. Question here? I'll try to look at everyone.
Just a basic question on fungi themselves. I never really, as I understand it, when a spore comes out, it lands someplace and it grows a mycelium, but it doesn't go anywhere unless it finds another spore from another mycelium and their DNA come together. Is that correct?
Well, in basidiomycetes, the stem and cap mushrooms,
Right.
that's generally true. They quickly form the dikaryon, where two different individuals meet, right?
Right.
And form that dikaryotic mycelium, two nuclei in each cell.
Yeah.
But in ascomycetes like Cordyceps, it's actually the monokaryon that is long-lived. So a spore may germinate and live as the monokaryon for the bulk of its life cycle.
All right.
Yeah, until it finds its host on an insect, incidentally. So that's another kind of mystery about going back to your question. Like two individuals find themselves together on an insect, and then they only form the dikaryon briefly when they form the fruiting body.
Is there some compatibility, like a male and female compatibility,
and
or
they
is
have
there
to have
more dimensions than
That's right. Yeah,
that?
they also have to have mating compatibility, so they have to be opposite mating types. So, yeah, yes,
All right.
good question. I think there was a hand up in the back earlier that I missed. This one?
Yes. When we were looking at the ascospores that were, 28 powers of 2,
Yeah.
like 32, 128. Is there 8 ascospores in each asci that are each that long?
Yes. Yeah, there are 8 ascospores in each ascus, typically, in all the Cordyceps. That's right. So eight of those long filamentous ascospores all next to each other like a bundle. Yeah.
So, follow-up question, I guess, how does the physics work that something so seemingly cumbersome can escape not only the ascus, but also the perithecia, and then actually like get into the wind or whatever? How does it travel so far?
Yeah, well, as they pass through the perithecial ostiole, the neck of the perithecium, they're squeezed. And they burst. The cap of the ascus bursts off and the ascospores come flying out of it. And sometimes you see them on a microscope slide as all together, where they've landed together. But yeah, it's under the pressure.
Ascus leaves through the ostiole?
Yes, yeah, the whole ascus comes out as it dehisces off the wall of the perithecium. Or sometimes they remain in the ascus and come back down into the perithecium if they're sticking. Yeah, sometimes they come in, sometimes they go out. Yeah, great question. Question here.
Your depth is so amazing. I wonder if you could talk about medicinal use of Cordyceps, and it seems like you have to be so selective which part of the organism you harvest, and is it species specific, the benefits versus potential toxins?
Great question. Yeah, so we're talking about medicinal use of Cordyceps, which I honestly don't know a tremendous amount about. There are some Cordyceps species that may be very medicinal, and I think that's actually there's some that have been used for centuries in herbal medicines. Then there's others that are being put forward as medicinal that may or may not be the same. But one of the take homes of my talk and maybe some of my research, hopefully, is that this is a huge group, a huge and ancient group with vastly different chemistries, and some newcomers are very often of the thought that Cordyceps are medicinal, right? So all Cordyceps are medicinal, but it's not at all like that, right? So it's not all Solanaceae are medicinal. Some of them are tomatoes, some of them are deadly nightshade, right? So, so as far as the medicinal ones, what you use, where you get it, I can't answer a lot of that. A lot of it's still just unknown, what is medicinal about Cordyceps sinensis or Ophiocordyceps sinensis, the Himalayan caterpillar fungus, and then some of the other ones that have been used. But yeah, it's really the Wild West, to be honest. There's a lot of unknowns and people doing experimental things with medicinal Cordyceps. So not a lot demonstrated in scientifically, sadly. Question here.
So in terms of finding them on leaves, is it like, are they more common on trees versus shrubs or certain species or anything like this?
Usually any leaf will do, but you gotta look on the underside. That's where,
Yeah.
where they will be. They'll get washed off the top, I guess. But if you look on the undersides of leaves, I like to flip things where you can get a lot of real estate. So grab a beech limb, flip the whole thing, and you got lots to look at pretty efficiently. So I find a lot of things under beech leaves, witch hobble, ferns are good, especially the longer lived leaves like ferns. And today we found a lot on mountain laurel. And so we flipped a lot of mountain laurel leaves. Yeah, rhododendrons are really great if you have those, down south especially. But usually it doesn't seem to matter what plant, it's just whatever plant. But sometimes there are certain insects that associate with certain plants. So in that case, it would be substrate specific. But yeah, so sometimes yes, sometimes no, sorry.
Thanks.
Question?
Just to clarify a statement you made, the Himalayan cordyceps, which have been used for centuries with vitality and other uses, is there any North American counterpart that you've uncovered so far?
There are somewhat.
Commercially.
Oh, commercially. It's sold commercially sometimes. There's some counterfeit fungi that are sold commercially as Cordyceps sinensis. One of the famous ones is the CS4 strain, Cordyceps sinensis strain 4, which was isolated from a Cordyceps sinensis in the Himalayas, which is a very difficult fungus to culture and grow in the laboratory. And what turns out what they actually had cultured was a secondary fungus, one of those other hyperparasites, a Cordyceps growing on a Cordyceps. So we now know that strain CS4 is something in another family, the family Cordycepitaceae, and it's in the genus Samsoniella, and it's not Ophiocordyceps sinensis. People actually still sell it as a medicinal mushroom, which I think is scandalous. Question?
Are these cyclic peptides that you are isolating from the cordyceps, are they toxic to normal human cells?
Yes. Yeah, those toxins from the Paracearia, those are toxic to humans, normal human cells too. We tested them on HUVEC cells, and yeah, they are actually more toxic to one of the cancer cell lines, which is promising. It could have a therapeutic window where we could kill cancer cells better, or maybe we'll learn how to target cancer cells. But they're very similar to other cancer drugs, so there's promise for the possibility of them to be therapeutic. I think we're out of time. Maybe I want to let us hear about Alaskan fungi too. And but if anyone has more questions, I'll be around at least through early tomorrow evening.